Hello everyone. Today we'll learn about polysaccharides. Most of the carbohydrates found in nature they occur as polysaccharides, and polysaccharides are also known as glycans. Polysaccharides are non-sugar in nature, which means that they are not sweet in taste. They are not sweet in taste. Whereas the monosaccharide and disaccharides are called as sugar. They are sugar in they are sugar, which means they are sweet in taste. Now, because like the polypeptide have they are synthesized on a mRNA templates, but the polysaccharide the synthesis is independent of the template. So that's why the number of monosaccharide units in a polysaccharide is not determined, it is not fixed. It is the intrinsic property of the enzyme or on like how much monosaccharides will be added to that particular polysaccharide. Therefore, they do not have a definite molecular weight. Now, polysaccharides are classified as homo and the heteropolysaccharides. Homopolysaccharide means they contain only one type of monomer. They are made up of only single type of monosaccharide units. Example: starch, glycogen, and the cellulose. Whereas the heteropolysaccharide, they contain more than one type of monomers. They may contain two or more than two monomers. Example include the peptidoglycan of the bacterial cell wall. Now the polysaccharide are there is one more way to classify the polysaccharide as the storage polysaccharide or the structural polysaccharide. It is depending. This classification is based basically on their structure on their function. So either they are doing the function of storage, starch in case of plants, and glycogen in case of animal. It is a storage polysaccharide. It means they serve the purpose of storage. Now, structural polysaccharide means they play the structural role as the cell wall or the extracellular matrix. So, chitin, it is chitin is present in the fungal cell wall and the exoskeleton of many insects. Peptidoglycan, it is present in the bacterial cell wall. Now we'll learn about one by one about these different polysaccharides. First of all, starch. Starch, as we all know, that it is a storage polysaccharide of plant. It means that the carbohydrate are stored in the plant in the form of starch. Now, they are mostly in the tubers like potato, sweet potato. So, they are the storage. Where is the that starch stored? It is stored in the plastids within the cell. Now, this starch they are present only in plants, and starch, as I've already told, it is a homopolysaccharide. It is a homopolysaccharide of D-glucose. D-glucose, and that's why it is called as glucan. What is a glucan? Glucan is a polysaccharide. A glucan is a term used for a polysaccharide which is composed only of glucose. Now, they are neutral in charge. There is no charge on this. Neutral in charge. Starch has two components. Starch has two components. One is amylose and another amylopectin. this is the minor component. It is the minor component comprising only 20%, equal to or nearly 20%. Amylopectin is the major component comprising of more than or equal to 80% of the starch. This is the water soluble part. This is water soluble. This is water insoluble in nature. This is a linear part. Linear unbranched. It means the sugar residues are linked in a linear fashion and there is no cross-linking. Whereas this is a branched polymer. The linear chains are often cross-linked at certain points like this. Now here the this glycosidic bond over here which links these two D-glucose residues is alpha 1 4. Similarly, here also in amylopectin, the the bond between the linear chain is alpha 1 4, but this cross linking, the bond over here in the cross linking, it is alpha 1 6. The this branching, the glycosidic bond at branching is alpha 1 6. It means that the one C1 of this glucose molecule is linked with the C6 of this residue. Now we'll see this detailed. Now this branching, how often this branching occurs. So this branching occurs nearly 25 to 30 residues after each 25 to 30 residues. Now just take a look at this. This is the amylose part, which is, as you can see, this is the linear component and the branching is alpha 1 4. The C1 of one this anomeric carbon is linked to the C4. So there is one reducing end and there is one non-reducing end in each chain. Now the branching, this will be the linear, the linear, you will see this branching at some point. So see at the branching, at the branch point, the glycosidic bond is alpha 1 6. The C1 is linked with the C6. Now this now if you see the structure, this is the amylose linear chains. The linear chains will be like this. So there will be one reducing end, there will be one non-reducing end per amylose chain. Now in case of amylopectin, this is the reducing end, and all other branch, this is the C4 end of all branch, because the C1 is involved in the glycosidic bond. So all these are the non-reducing end. All of these are the non-reducing end. So in case of amylopectin, there is one reducing end and all other are non-reducing end. There are as many as non-reducing end as many there are branches. As many branches are there, so that there will be that much reducing end. But there will be only one reducing end per amylopectin. So in starch, it is the amylose which is responsible for the iodine test. The amylose, as we have seen in the previous slide, it is a linear polymer and it exists as a right-handed alpha helix. Right-handed alpha helix. So this core of this amylose is of the exactly of the right dimension to accommodate the iodine in the form of iodide ion, and therefore it gives a blue black color. This is a test which is used for the starch identification, as in which the compound is just mixed with the iodine, few drops of with iodine, and if it gives a blue black color, it indicates that it is the starch. Now our next polysaccharide is dextrine. Dextrine gives a violet color compared to the starch when tested with iodine. It is also a polymer of D-glucose. Dextrine is formed due to the partial hydrolysis of starch. What is dextrine and how it is formed? When starch is hydrolyzed either with heat or the enzyme amylase, or in the presence of dilute HCl, then this is hydrolyzed to smaller product that is called as dextrine. So dextrine is actually a smaller form of starch, and there will be more generation of the reducing end, so that's why it is more reducing. That's why it is more reducing than starch. Because hydrolysis of starch will lead to smaller product, and thus there will be generation of more reducing ends. That's why it is more reducing than the starch, but less reducing than the monosaccharides and the disaccharide. It is soluble in water, and it is used as adhesives. So next, one more polysaccharide is aniline. It is a plant storage polysaccharide like the starch, and it is a fructan. So till now we have heard about or learned about glucan. So here it is a fructan. It means it is a polymer of D-fruct D-fructose D-fructose, and it is also a linear polymer. It is a linear, that is, there is no branching. So it is a linear unbranched polymer, and the glycosidic bond present between two fructose unit is 2,3 beta 2,3 glycosidic bond. It means that C2 of one fructose, C2 of one fructose is linked with the C3, and the configuration of this beta and the C2 is beta. So this is all about inulin. Now we'll move to next to glycogen. Next is glycogen. Now we all know that glycogen is a storage polysaccharide of animal, and where it is synthesized. So it is synthesized and it is synthesized in liver and skeletal muscle and stored there. So the amount is higher in muscle. The amount of glycogen is higher. Amount is higher in muscle, and in liver the density is higher. That is, the concentration density is higher. So glycogen is stored and synthesized in the liver, in the hepatocytes, in the it is synthesized in the and it is stored in the cytosol. It is synthesized and stored in the cytosol of the hepatocyte and the cytoplasm of the skeletal muscle. Now it is a storage polysaccharide of animals. It is also a homopolysaccharide. Homopolysaccharide means composed of only one monomer. So it is a homopolymer of glucose. That's why it is also a glucan. It is also a glucan. Now this is Glycogen is also present in animals as well as fungi. Animals as well as fungi. Uh, this is also a branched polymer. Branched polymer like amylopectin. The branching in glycogen is like amylopectin. Amylopectin. So, as in amylopectin, the linear cross linkage is alpha 1. The linkage between the linear chain is alpha 1. The glycosidic bond in linear chain is alpha 1, alpha 1 for glycosidic. And at the branch point, the glycosidic bond is alpha 1, 6. Alpha 1, 6 glycosidic bond. Now, like this, you can see over here also. This is C1 of this is linked to the C4 of this. Similarly over here. So the linear branch are linked in alpha 1 4 fashion. Now the branching at the cross glycosidic bond at the branch point is alpha 1 6. So C1 of 1 is linked with the C6 of another sugar. This is how this branching occurs. Now, like the amylopectin, there will be one reducing end. There will be only one reducing end over here. This is the C1. This is the reducing end. All other these end up in C4. All other because the linkage over here is 1 4. The linkage between these two is 1 4. So this is the C4 carbon. So wherever the ends of the branching chain will end in C4. All these will be the non-reducing end. There will be one and only one reducing end, like the, and all other are non-reducing. That's why this is a non-reducing sugar. Now, why don't we? The question is, why don't we store glucose, and why do we need to? Why it is needed first to convert the glucose to glycogen, and and why then it has to be stored within the liver and the muscle cells. So, why glucose is not stored instead of the glycogen? So, the storage of glycogen, it is equivalent to it is equivalent to a glucose concentration of 0.4 molar. If 0.4 molar glucose is stored within the cytosol, if the glucose is stored if glucose is stored within the cytosol, then up to a concentration of 0.4 molar, the osmolarity, because the solute concentration is high enough, so obviously the osmolarity will go high. Osmolarity. Osmolarity will be very high, and the solute concentration is high inside. It is high inside and low outside, so there will be movement of water inside. It will lead into swelling of this cell and the cell lysis. Uh, Glycogen is insoluble and therefore it contributes very little to the osmolarity of the cytosol. That's why it is preferable, or it is by evolutionary, it is preferable to store glycogen and not glucose. There is one more reason why the glucose is stored, because the if if glucose would have been stored, the concentration to up to a concentration of zero point 0.4 molar, and the concentration of glucose outside is 5 millimolar. So the uptake of glucose, the free energy change required to uptake or required for the uptake of glucose against this, and this gradient will be very high. So it will be prohibitively very large, and the glucose uptake would not have taken against it. The energy required to uptake this glucose against this energy gradient would be very high. So energetically it would not be favorable to transport this glucose from a lower concentration to a concentration of zero point 0.4 molar. Zero point 0.4 molar is actually 400 millimolar. So you can see the difference in the concentration across the cell membrane. So the energy change that would be required to transport this glucose from the here to here would be very high. It would not be, it would be prohibitively very large. So that's the reason for that glucose is stored as glycogen and not in the form of glucose. Now another polysaccharide is dextran. So dextran are basically, they are the bacterial and the yeast polysaccharide. They are also glucan, and they are made up of they are glucan. So it is a linear polymer. It is a polymer of D-glucose. The linkages, linkage between the linear chain is alpha 1,6, whereas there are branches, and the branching is at usually at 1,3, but the branching could also be uh, at alpha. And here the linkage in the linear chain, they are linearly joined by alpha 1,6, and there could be branching by usually by alpha 1,3, but they may also contain, the branching could also be due at alpha 1,2 or alpha 1. The dextran, the dental plug, they're actually formed by the bacteria growing on the surface of teeth, is rich in the dextran. Synthetic dextran uh, that are used in protein purification in the size exclusion chromatography, they are actually the commercial product, or they're actually chemically synthesized dextran known as the Cefadex. They are of different porosity and are used in protein uh, purification or the fractionation of protein by the size exclusion chromatography. So if you have, before moving to the next slide, please do subscribe, and if you haven't subscribed yet, and please do share it with your friends, your batchmates, for whom you could think it could be useful. Next polysaccharide is cellulose. As we all know, cellulose is a plant, is a polysaccharide present in plant cell wall. It is fibrous, it is tough, it provides strength to the plant cell, and then it is also water resistant. It is a linear unbranched homopolysaccharide of D-glucose. That's why it is also a glucan. And it composed of, the cellulose is composed of nearly 10 to 15,000 molecules of D-glucose. It is a homopolysaccharide. It is a homopolysaccharide of the glucose, a linear chain, it is a linear chain, and the glycosidic bond present between connecting two glucose molecule is beta 1 4. It is the glycosidic bond. The glycosidic bond between two glucose molecule is beta 1 4. Now they are arranged in a linear fashion, so there will be one reducing end, and each chain will have one reducing end and one non-reducing end. So now because these linear chains, arranged, the cellulose chain, they are arranged parallelly, nearly 40 cellulose chain, 30 to 40 cellulose chain, nearly 30 to 40 cellulose chains arranged parallelly to form microfibril. Microfibril. Now this cellulose, it cannot be digested by animals. Animals do not have enzyme. Animals do not have this enzyme cellulase, which is required for the hydrolysis of this bond and thus digestion of cellulose. So this cellulase is produced by some bacteria, some protists, and wood rot fungi. Wood rot fungi. This bacteria, okay, the cattles which feed on grass, the cattle, in the gut of cattle there lives a symbiotic bacteria. There is a bacteria which lives in symbiotic relationship with the cattle. Bacteria which can secrete this cellulase. It secretes cellulase enzyme. It secretes cellulase enzyme which digests the cellulose present in the grass or the plant product eaten by the cattle. Similarly, this termites, the termites which feed on which eat wood. So in the gut of termites there is a protist called as trichonymph. Trichonymph. There is a protist known as trichonymph which is present in the gut of this termite, which produces cellulase and thus helps cellu it produces cellulase enzyme which digests the cellulose in the wood. Now next polysaccharide is chitin. Chitin is present in the fungal cell wall and the exoskeleton of some arthropods. Exoskeleton of some arthropods like insects, crabs, lobsters, etc. So they have this tough exoskeleton which is water resistant. It is a structural polysaccharide and it is a homopolysaccharide. It is a homopolysaccharide. It means it is made up of only one type of monomer, and that monomer is glucnac. Glucnac. Glucnac is N-acetyl glucosamine. So this is a derivative of glucose where the at the second carbon, the OH of the second carbon is modified to acetylated amine. So it is a linear, chitin is a linear polymer of glucan, linear or it is or unbranched. So the glycosidic bond present over here is also beta 1. The glycosidic bond present in the chitin molecule is beta 1. And then it it also extend to form fibers. These arranged into form fibers. The fibers could be parallel or anti-parallel, or it could be a mix of parallel and the anti-parallel. Please do not forget to subscribe and share it with your friends. Thank you.